Hello and welcome to the HIVRNAtestguide.com YouTube channel, your online partner for quick, affordable, and 100% confidential HIV testing. Whether you're looking for quick, confidential results or simply want to stay informed about the latest in HIV research, you're in the right place. For more information, visit HIVRNAtestguide.com. Check the link in the description below or visit the bio section of our channel. You can access our more than 4,500 HIV testing labs across the United States, just a click away. So, without further delay, let's start today's video.
Today, we are focusing on a topic that has, for so long, felt like one of the most insurmountable challenges in global health, the HIV pandemic.
And for decades, I mean literally for decades, the search for a truly effective vaccine has been one of science's most profound and frankly frustrating quests. The virus is just a master of biological evasion.
Right.
However, our sources today, and they're all detailing the IAVI G001 trial, they point to something entirely different, something new. We're looking at a radical new vaccine strategy that suggests we might finally be witnessing the first major foundational step toward ending the HIV crisis as we know it.
That shift in tone alone is, well, it's seismic, isn't it? Given the history of failed attempts. So the big question we're wrestling with today is, are we finally close to having an HIV cure? Or maybe more accurately, a universally effective preventative vaccine? We've analyzed the sources on this IAVI G001 trial, and you're right, it's not just an incremental improvement. It feels like a wholesale rethinking of how we engineer immunity against these really complex pathogens.
Precisely, and that's our mission for this deep dive, to strip away the high-level headlines and really get down to the mechanics of it. We need to understand the strategy they're calling germline targeting. We need to understand why it represents such a brilliant piece of molecular engineering and what that almost unbelievable 97% immune response rate truly validates for the future of global medicine.
Okay, let's unpack this, because I think to really grasp the scope of this breakthrough, you have to understand the specific target they were aiming for. We're not talking about just simple protection here. We're talking about designing immunity at the cellular level, and it's all focused on what scientists call the holy grail of HIV prevention, broadly neutralizing antibodies, or bNAbs, as they're known.
These are absolutely the immune system's elite weapons. To put it simply, most antibodies your body produces, say after a flu shot, are highly specific. They're designed to recognize one particular strain of a virus.
Okay.
And if that virus mutates even slightly, that antibody can become useless. HIV, it thrives on this specificity.
So if the immune system is like a sophisticated security force, a regular antibody is like a guard who's been trained to recognize just one specific face on a wanted poster.
That's a good way to start, a really good analogy. Now compare that to bNAbs. They are broadly neutralizing because they recognize a whole array of villains all at the same time. They target these conserved, crucial sites on the virus parts that are necessary for the virus's survival. And that means they work against multiple variants of HIV at the same time.
Wow.
The entire scientific goal here isn't just to find a bNAb, it's to teach the human body how to manufacture these incredibly powerful universal master keys on demand.
To truly appreciate the elegance of the IAVI G001 approach, then, we have to understand the decades of frustration that came before it. Why has HIV been so uniquely difficult to combat with traditional vaccines?
It's a great question because the history of HIV vaccine development is, frankly, it's littered with failures. Standard approaches that worked beautifully for diseases like polio or measles, they just couldn't get a foothold against HIV.
So what's the fundamental difference? Why the struggle?
The challenge is really rooted in the virus's molecular structure and its sheer speed of mutation. When HIV enters the body, it uses this large, complex surface protein called the NV trimer. You can think of it like a rapidly spinning armored shield that it uses to attach to and infect our immune cells. And this NV protein is highly variable.
So it's not one target. It's like trying to shoot at 1,000 different shapes that are appearing almost instantaneously.
Precisely. The sources all highlight that the core difficulty lies in HIV's ability to constantly mutate and present multiple diverse variants. They're known as quasi-species. And this can happen even within a single infected person. It's like trying to hit a target that's changing its size, its speed, and its location every few milliseconds.
And standard vaccines, what do?
Standard vaccines often elicit a strong response, but it's targeted at the variable regions of that N protein, the outer shell, the parts that change rapidly. So those antibodies become obsolete almost immediately.
Which means the antibodies we do make are often just ineffective decoys. They distract the immune system from the real fight. That's why we need a response that's, I guess, smarter than the virus itself.
Exactly. And this brings us right back to the structural necessity of bNAbs. The key is their ability to cross-react because they target the conserved regions of the virus. This is really the heart of the matter.
The conserved regions, what does that mean exactly?
So imagine that spinning ENVO trimer shield we talked about. While the outer surface changes constantly, the virus still needs certain molecular structures, these specific functional regions, to remain unchanged. They have to stay the same because they are absolutely essential for the virus to bind to human cells and replicate.
Okay, so if the virus is changing its armor every single day, the conserved region is like the one critical screw holding the whole machine together. The virus cannot afford to mutate that part, or it literally loses its ability to function.
That's exactly the biological constraint we're looking at. The problem is that conserved region is often buried or shielded by sugars and other molecular clutter until the exact moment of infection. Previous vaccines failed because they couldn't present this buried, essential target to the immune system in a way that would train the body to see and attack it. They were only showing the immune system the rapidly changing outer shell.
So the scientific breakthrough here is purely structural then. It's about engineering an immunogen, the vaccine component, that strips away all the distracting variable noise and only presents that conserved critical structure.
That's the first hurdle they had to overcome. And if we can succeed in making bNAbs, the clinical implication is just monumental. A vaccine regimen based on successfully inducing these specific antibodies could potentially offer true, long-lasting protection against the entire global diversity of HIV strains, regardless of regional variations. It turns a localized defense into a global offensive strategy.
But, and this is the crucial puzzle piece the sources explain, why doesn't the body just start making these bNAbs naturally, especially after prolonged exposure to the virus itself?
It's a fantastic question. And the reason is because the starting materials, the specific B cells that have the potential to become bNAb producers, are extremely, extremely rare. Our immune system uses a kind of biological lottery, it's called VDJ recombination, to generate this huge library of B cell receptors. And only about 1 in a million B cells might have the right foundational genetic blueprint, the germline configuration, to even begin the process of becoming a bNAb producer.
1 in a million. Wow, that is a needle in a massive haystack.
It is. And here's the problem. When a person is naturally infected, the viral chaos is so fast and so overwhelming that the common, easily activated B cells respond first. These common B cells produce weak, non-neutralizing antibodies that just dominate the response, and they essentially push those rare, high-potential germline B cells to the side. So those rare high-potential cells never get the specific clean signal they need to begin their sophisticated training process.
So the body has the potential. The genetic blueprint is there. It's just in a tiny fraction of cells. But the natural infection process is too messy, too disorganized to activate the right troops. We need a way to bypass all that chaos and strategically intervene to coach only those rare specialists.
That is the ultimate goal of the IAVI G001 trial: targeted recruitment and initial training.
And here's where it gets really, really interesting, because this trial is fundamentally about vaccine engineering precision. They aren't trying to fight the virus with the traditional broad antigens. They are selectively hunting for those 1 in a million cells. This is the concept of germline targeting.
Germline targeting is, to put it simply, a highly focused search and activate mission. We mentioned the B cells start life with a foundational genetic blueprint, the germline configuration. This is their genetic starting line. The majority of B cells, when they're activated by a natural infection, they'll develop into common antibody producers based on common germline blueprints.
And germline targeting is specifically designed to ignore all those common recruits and only focus on the ones with that high potential blueprint.
Exactly. They are searching for the B cells whose receptors are structured at the germline level to interact with that highly conserved critical site on the HIV-NV protein. The vaccine is designed to activate only those rare B cells, the ones that possess the necessary molecular structure that, after future refinement, will become the universal bNAbs. The IAVI G001 trial is defined as that crucial priming step, just initiating the process in human volunteers.
That has to require an incredibly sophisticated molecular tool, though. So how did the researchers design something that could selectively find and activate these rare cells without triggering a distracting general immune response?
They created a custom-built, highly engineered structure. It's called the EOD-GT8 6-dimer immunogen. This is the specialized tool, and its design is really the core of the entire breakthrough. An immunogen, again, is just the part of the vaccine that stimulates the immune response.
That 6-dimer designation, that's a huge clue to the engineering, isn't it? It suggests it's not just one single protein floating around.
It's vital. The EOD-GT8 part refers to the specific protein component, which mimics that conserved binding site of HIV. The 6-dimer part signifies that this component is assembled into a geometrically precise nanoparticle made up of 60 identical copies of the protein, all arranged in this repeating structure.
60? Why 60? Why that level of, what do they call it, multivalent complexity? Why not just 1 or 2 copies?
That is the key insight from Scripps Research and the designing labs. When a B cell, especially a rare one, recognizes an antigen, the immunogen, it doesn't happen with a single molecular handshake. The B cell receptor needs to be cross-linked. It needs to bind to multiple copies of the antigen at the same time to generate a powerful activation signal.
I see.
And because those rare germline B cells only bind weakly to the first antigen, remember they're only precursors, you need a really high concentration of target sites presented perfectly in 3 dimensions to force that strong cross-linking signal.
So the 6-dimer nanoparticle acts like a molecular bullseye. It increases the probability exponentially that the rare B cells will bind and then receive the signal to activate. It's like turning up the volume on that encrypted message we talked about earlier so that the Special Forces recruits, and only them, can hear it over all the noise.
Precisely. That nanoparticle structure provides the necessary multivalency to achieve that critical B cell activation. And its function is twofold, as detailed in the sources. First, it selectively activates the rare B cells with the right germline configuration. And second, it guides them in the direction needed to mature.
And that guidance part is setting the stage for the next crucial immunological process, which I imagine is far more complex than just simple activation. It's forcing them into a genetic refinement process.
Absolutely. The activation of these precursors is merely step 1. These activated B cells will now enter the germinal centers of the immune system, and that's where they'll begin a targeted process called somatic hypermutation. They start mutating their own DNA to refine their antibody structure, making the resulting antibody bind more broadly and more powerfully to the actual NV protein. The EOD-GT8 6-dimer is the perfect starting template. It sets that evolutionary process off in exactly the right direction.
It's the ultimate example of advanced preventative architecture, the shift from trying to block the virus after it mutates to preemptively training the immune system with precision engineering to create a future master key. This feels like a fundamental game changer for complex pathogen vaccine development globally.
It is.
So, with all that brilliant, precise science informing the design, the IAVI G001 trial became the critical test. Could this engineered immunogen, this 6-dimer nanoparticle, actually achieve this incredibly targeted priming within the complexity and diversity of the living human immune system?
And the results, they confirmed the biological model with staggering efficacy. This was a phase I clinical trial, which is crucial to define. Phase I trials are primarily focused on 2 things. First is safety, and second, for a novel approach like this, is mechanism validation.
Right, so they're not yet looking to see if the patient is protected from infection. They're just asking, did the specific molecular intervention we designed work as we predicted it would in humans?
Exactly. The trial was rigorous. It involved 48 healthy volunteers recruited and monitored at research centers in Washington, D.C. and Seattle, and the focus was squarely on measuring that initial immunological response.
And the primary result exceeded expectations.
It did. Dive into the data. 97% of participants developed a special kind of antibody response that was designed to block HIV. When a phase I trial achieves a 97% success rate for mechanism validation, especially against a target as evasive as HIV, it represents a profound biological consensus.
That percentage doesn't just confirm the success of the germline targeting approach, it confirms the surgical precision of the EOD-GT8 6-dimer. It proves the molecular key worked, well, universally across nearly every human immune system they tested. It successfully unlocked the potential of those rare B cells.
And we absolutely must emphasize the safety profile, which is so critical for phase I. The sources were absolutely clear on this. The participants' bodies responded exactly as researchers hoped, creating that highly specific, correct antibody response without any serious adverse effects. Achieving such high efficacy in a novel approach while maintaining a benign safety profile, that is the ideal green light for accelerating into the subsequent phases.
Let's get even more granular on the data, though. How did researchers quantify the success of the priming? It's one thing to say they responded, but how did they actually count those rare 1 in a million cells?
This is where the deep dive into specialized assays really confirms the rigor of the science. Researchers used these sophisticated cell sorting techniques, like flow cytometry, which allowed them to isolate, identify, and physically count the specific B cells that had successfully bound to the 60-mer immunogen.
So they didn't just guess the response, they visually confirmed and quantified the activation.
Exactly. Our sources reveal that, on average, each person activated an impressive count of 30 to 65 bNAb precursor B cells. Now think about the biological context of that number. Given that these are cells with a 1 in a million chance of activation under natural circumstances, to programmatically activate 30 to 65 of them per person is a monumental biological achievement.
So if the goal was recruitment, they not only recruited, but they achieved a critical mass of recruits that are now ready for specialized training. That concentration of potential is what determines whether the next phase of the vaccine regimen can actually succeed, right?
It gives scientists the necessary confidence. This concentration is the quantifiable proof that the immune system has been redirected, that it's bypassing the clutter of common antibodies and is now focusing on the foundational, conserved elements of HIV. It sets a very strong basis for the multi-step maturation process that has to follow.
And the researchers also had to ensure a crucial element of quality control. They needed to know they weren't accidentally activating the wrong cells. B cells that might develop into antibodies that bind weakly, or even worse, generate antibodies that actually help the virus shield itself.
That is vital. Activating off-target cells could lead to a diluted, ineffective, or even a harmful immune response later in the regimen. So the secondary validation performed by the researchers was specifically designed to screen for this.
And what did that quality check reveal about the activated precursors?
The sources confirmed the ideal scenario. The bNAb-producing cells that were activated turned out to be the strongest and most effective ones, the highest potential candidates. This just confirms the astounding precision of the EOD-GT8 60-mer design. It wasn't just a magnet for rare cells. It filtered for the most high-fidelity, high-potential rare cells available. This robust foundation is what allows the entire field to move forward with such velocity.
This kind of monumental scientific leap, conquering a problem that has defied scientists for 40 years, it's never the work of a single lab. And looking at our sources, the scale of the collaboration required here is immense. It's bridging academia, nonprofits, and the pharmaceutical industry.
It truly is a testament to the power of cross-institutional scientific commitment. You see a confluence of specialized expertise. You have Scripps research, which drove much of the foundational molecular design and the antibody engineering. Then you have IAVI, the International AIDS Vaccine Initiative, which was instrumental in designing, funding, and managing the critical human trials. And then on top of that, you have massive institutional support from the NIH, Fred Hutch, GSK, and crucially, Moderna.
That collaboration model is essential, isn't it? Because you need the theoretical molecular precision from a place like Scripps, the clinical rigor of IAVI and Fred Hutch, and then the ability to manufacture and iterate at speed, which is where Moderna and GSK come in. It's a unified global effort dedicated to solving a global problem.
And that link to Moderna leads directly into the next phase of acceleration. The sources highlight the massive technological shift that has taken place globally in just the last few years, allowing this HIV strategy to piggyback on established success.
The mRNA accelerator.
Exactly. The new HIV vaccine strategy now has its own parallel development path using the mRNA platform. And this is a profound game changer for the subsequent steps of the immunization regimen.
Why is mRNA particularly crucial for this multi-step germline targeting approach? What does it enable?
Because germline targeting doesn't end with that 60-mer primer. As we discussed, that primer activates the rare B cells. That's step 1. Now, scientists need a step 2, a step 3, and perhaps even a step 4. They need a series of distinct, slightly modified immunogens delivered sequentially to guide those 30 to 65 precursor cells through the maturation process, refining their antibodies through that somatic hypermutation we mentioned.
So they need to quickly design and test multiple different follow-up immunogens, each one slightly different, to progressively coach the B cells toward broader neutralizing power.
That's where mRNA technology shines. Historically, producing large quantities of multiple, complex, customized protein immunogens, like the EOD-GT8 60-mer, was incredibly time-consuming and expensive. If step 2 didn't work, you might spend months or even a year just manufacturing the next candidate to try. With mRNA, you are only changing the genetic code. A sequence of letters and production can be scaled and adapted almost instantly.
So this means the refinement process, the process of turning the potential of those 30 to 65 precursor cells into fully operational, robust bNAb producers, can be tested and iterated in months, not years. That speed is just paramount in overcoming a challenge that has stalled for decades.
And the sources confirm that this acceleration is already manifesting in the global reach of the next steps. The mRNA version is already undergoing subsequent trials, reflecting the global focus and the need to test the entire immunization strategy against diverse viral populations.
Where are these trials strategically located?
They're taking place not only in the U.S., but critically, in regions that bear the brunt of the pandemic. We see trials underway in Rwanda and South Africa.
That inclusion is so vital, from both an ethical and a scientific perspective. Scientifically, it ensures that the subsequent steps of the vaccine regimen, the steps that induce the full bNAb function, are validated against the widest possible array of viral variants common across sub-Saharan Africa. And then, ethically, it ensures that the solution is validated precisely where the need is greatest, reinforcing that the bNAb goal is true, broad, and global protection for all populations. This is really the global promise of this technology.
So what does this all mean for you, the person listening? The IAVI G001 trial represents a definitive molecular success. It successfully demonstrated that germline targeting is a viable, high-efficacy strategy to initiate the production of crucial bNAb precursors in humans. We have solved the recruitment problem. We've set the stage.
We have the foundation upon which an entirely new generation of HIV vaccines can be built. And our sources are extremely hopeful. The data gives us real hope that the next few years may finally bring the world's first truly effective, globally protective HIV vaccine. This is a monumental moment in immunology. We're pivoting from hoping for a response to actively engineering one with surgical precision.
We have confirmed the existence and the activation of those specialized trainees, those 30 to 65 bNAb precursor B cells, and we successfully guided them into initial specialized training using that highly engineered EOD GT860mer.
Which brings us to a final provocative thought for you to consider as you digest this massive breakthrough. If the priming step, the IAVI G001 success, is now complete, what is the immediate looming challenge for scientists right now?
The challenge shifts entirely to the maturation process, and the complexity there is immense. A full vaccine regimen will require what is called sequential immunization. This is where the difficulty really lies now. We need step 2, step 3, and step 4 immunogens that are distinct enough from the primer to continue pushing those precursor B cells to mutate their own genes, that somatic hypermutation, to increase their binding strength and their breadth.
So the B cell's activated, but now it needs to continuously evolve itself, right? It can't stay static.
Exactly. The B cells are in a race against time, needing to continually refine their receptors to become masters of recognition. This is the difference between having a specialist trainee and having a fully operational, battle-ready operative that can neutralize any HIV variant it encounters. The ongoing multi-stage work, using the mRNA platform to rapidly test and deploy the subsequent immunogens designed to push this affinity maturation, that is what truly determines how quickly we achieve that goal of a truly effective vaccine. The hard work of guiding evolution is just beginning.
That is a fascinating pivot. Acknowledging the triumph of step 1, while recognizing the sheer biological complexity of the path ahead. We're watching vaccine science evolve from generalized targeting to precise molecular coaching, and it's just electrifying to watch.
A truly exciting chapter in human health.
And as we move into a hopeful new chapter for HIV prevention, our sources consistently emphasize the ongoing need for public health action and awareness. We want to share a resource highlighted in your material that provides a valuable, confidential service. If you are looking for quick, affordable, and confidential HIV testing anywhere in the United States, we encourage you to visit HIVRNAtestguide.com.
Take care of yourselves, stay informed about this unfolding story of scientific hope, and please share this knowledge widely.